Hello, this is Yashvin. In this video, I'll be solving paper 2 waiting to October, November 2020 and 700 biology. 1. Figure 1.1 is a photomicrograph of a transverse section through the stem of a creeping buttercup named Ranunculus repens, where you are given the name of the genus and the species. A. Draw one label line on figure 1.1 to indicate one precise location where phloem sieve tubes occur. So all around the center of the stem, you've got these vascular bundles. The top region, which is shaded in this photomicrograph, would be the phloem. You can label one from any of the vascular bundles. B. State the role of phloem sieve tubes. So these sieve tubes transport assimilates, which are basically substances like sucrose and amino acids dissolved in phloem sap, and they are transported from source to sink. So where source would be the area where they are produced or where they have been stored, and sink would be the area where the assimilates would be used up. See, a good quality plant diagram of figure 1.1 will show only the tissues in the stem in their correct location and in correct proportions. 1. State the name of the tissues. The tissue that forms the outer layer of the stem section of R. repens in figure 1.1. The outer layer of tissue is formed by epidermis tissue. D. Draw a diagram in the space provided to show the structure of a typical plant cell. Label your diagram with the name of any structures that are found only in plant cells. Do not label any structures that are also found in animal cells. Alright, so here I've drawn a cell wall, a chloroplast indicated by the fact that it's got thylakoid membrane-like structure, a tonoplast, the membrane surrounding vacuole, and a large vacuole present in the center of the cell, and a starch grain, which acts as a storage. Starch granule it should be, and a starch granule which acts as a storage. You can also draw a plasmodesmata, but since this question is only of 5 marks and I've drawn 5 structures, that will be all I'm drawing over here. 2. People who smoke tobacco are at an increased risk of developing non-infectious diseases of gas exchange system and the cardiovascular system. In 2004, a study was carried out on the most popular brands of filter cigarettes from each of the 6 World Health Organization regions. In this study, the smoke passing through the filter was analyzed for the tar, nicotine, and carbon monoxide content for each brand of cigarette. For each WHO region, the main content of tar, nicotine, and carbon monoxide of the different brands of cigarette was calculated, and the results are shown in figure 2.1, which is a bar chart. And here you have got the key to WHO regions, each with an acronym, where the darkest shaded bars represent amount of tar, nicotine is the lightest shade, and carbon monoxide is this grayish shade. A. With reference to figure 2.1, list the 2 WHO regions where smokers are at the highest risk of developing diseases of the gas exchange or the cardiovascular system. Gas exchange diseases are caused by tar, and cardiovascular diseases would be caused by carbon monoxide and nicotine. And so the 2 regions where smokers have highest risk of these 2 kinds of diseases would have to be living in an area or 2 areas where cigarette smokes contain the greatest amount of tar, carbon monoxide, and nicotine. So the first region is here 0, which has the greatest amount of tar, where cigarettes contain the greatest amount of carbon monoxide and nicotine. B. With reference to figure 2.1, explain why smoking a popular brand of cigarette from Afro region is more likely to increase the risk of blood clots forming than smoking a brand of cigarette from Euro region. The component of cigarette smoke which increases the risk of blood clots is nicotine, and if a brand from Afro is more likely to result in blood clots, that means it has a greater proportion of nicotine, which you would probably able to see over here by comparing it with the Euro region. And as predicted, the Afro cigarette contains greater proportion of nicotine than that in Euro. As the question asks for reference, you would get 1 mark for writing that the Afro region cigarette contains greater amount of nicotine. You'd also have to quote some data. So here I've mentioned the amount of nicotine in each of the 2 brands. C. One short-term effect of cigarette smoking is a decrease in the supply of oxygen to body tissues. Describe and explain why cigarette smoking leads to a decrease in supply of body tissues. Cigarette smoke contains carbon monoxide, which binds irreversibly with hemoglobin, and so it prevents the binding of oxygen with hemoglobin in the lungs, which increases the percentage saturation of hemoglobin with oxygen, and therefore that means lower amount of oxygen is unloaded at respiring tissues, and therefore their supply of oxygen is decreased. Three, a red blood cell goes through a number of stages in the bone marrow before it is released into the circulation to carry out its role of oxygen transport. Figure 3.1 is a transmission electron micrograph of developing red blood cells in the bone marrow. Each cell is known as a erythroblast. A figure 3.1 shows the erythroblasts at a higher magnification than can be obtained using the light microscope, meaning that figure 3.1 is obtained using an electron microscope. Explain the advantages of using an electron microscope to obtain images such as that in figure 3.1. The resolution of a light microscope is only 200 nanometers, which is much larger than the size of a red blood cells, meaning that they can only be viewed using an electron microscope, as it has a much higher resolution of 0.5 nanometers instead, as opposed to the 200 nanometers of light microscope. B. Describe the differences between a erythroblast and a mature red blood cells. So erythroblast would not have yet developed any features of a typical red blood cell, and so you're supposed to mention the features which distinguish a red blood cell from a regular animal cell. A red blood cell would have a biconcave shape. Also, it would have no nucleus, since that increases the space for packing maximum number of hemoglobin molecules. It would also lack a mitochondria. Since erythroblast has not yet matured into red blood cells, it would have all of those features. It would have a nucleus, it would have a mitochondria, and it would not have a biconcave shape. Have a nucleus, it would have a mitochondria, and it would not have a biconcave shape. C. The maturing red blood cell synthesizes hemoglobin and other proteins. One, complete figure 3.1 to name 3 organelles or cell structures that are involved in the synthesis of a fully functioning protein. State one way in which the named organelle is involved in protein synthesis. Well, first off, proteins need a code or a length of DNA that codes for the sequence of amino acids in the polypeptides, and that would be the gene present in a nucleus. So the organelle we stating first would be nucleus, and it's used to transcribe the mRNA using the genetic code. So it is required for transcription process of the protein synthesis. Next up we have a translation which takes place in the ribosomes. And last but not least, all of these polypeptides or proteins are modified in the Golgi body. This could be, for example, formation of disulfide or hydrogen bonds. 2. Figure 3.2 shows a reversible reaction that takes place within red blood cells, and you're supposed to name the protein that catalyzes the reaction. That would be carbonic anhydrase. The SPTB gene codes for a protein that provides stability and support to the cell surface membrane of the red blood cell. Mutation in SPTB result in red blood cells that are spherical in shape. This mutation leads to a disease known as spherocytosis type 2. D suggests why a person with spherocytosis type 2 has a reduced ability to take up oxygen in the pulmonary capillaries compared with a person who has normal red blood cells. One of the key features of a red blood cells is its high surface area to volume ratio. What that does is not only does it increase the surface area for diffusion of gases like oxygen and carbon dioxide into and out of the cell, it also reduces the distance that these gases have to travel, and particularly oxygen has to travel from the alveoli into the red blood cells to reach the hemoglobin. Now, because this mutation leads to the red blood cell having a spherical shape instead of the biconcave, the mutated gene results in those features being absent, and so this reduces the surface area to volume ratio of the red blood cells and increases the distance that an oxygen molecule has to travel before reaching the hemoglobin molecule in the red blood cell. E. Increases in the water potential of the blood plasma have greater effects on the red blood cells in people with spherocytosis type 2 than on normal red blood cells. Compare and explain the effects of an increase in water potential of blood plasma on spherocytosis type 2 red blood cells and normal red blood cells. Well, first off, you're supposed to explain what happens as a result of water potential, and then you'll be getting the rest of the marks for comparing them. If the water potential of blood plasma increases, that means water would move down the water potential gradient by osmosis into the red blood cell, and eventually the red blood cell would burst since it does not have a support like cell wall. Now, for a regular red blood cell, since the shape is biconcave, it takes a greater amount of water for it to swell and then burst, whereas a red blood cell for a person suffering from spherocytosis is already swollen, and so it doesn't need as large of an amount of water for it to burst, as it would already be swollen, and that is how you'll have to compare the two cases. For carbohydrates, proteins, and nucleic acids are types of biological molecules. Within each type, there are examples of molecules that can be described as polymers. A polymer is a large molecule composed of smaller repeating molecules known as monomers. Complete table 4.1 to produce a summary of some named biological polymers. Alright, so starting with amylose, the consequent monomer is glucose, and the type of bond between monomers is a glycosidic bond. This bond is present between glucose molecules in amylose, amylopectin, cellulose, and glycogen as well. So once again, we have a glycosidic bond in here. Collagen's monomer are amino acids, since collagen is a protein that are linked together by peptide bonds, DNA has DNA nucleotides as its monomers with phosphodiester bonds between adjacent nucleotides, and glycogen has glucose molecules for its monomers. B. Explain why an amylose molecule and a cellulose molecule have very different structures even though they both have glucose as the constituent monomer. That is because amylose is made of only alpha glucose molecules joined by 1,4-glycosidic bonds, whereas cellulose is made up of beta glucose molecules, and so every alternate molecule has to be rotated 180 degrees to form the 1,4-glycosidic bonds. C. Polymerization of DNA nucleotides occurs during the semi-conservative replication of a molecule of DNA during interphase. Describe the semi-conservative replication of DNA. For DNA to be replicated, the two strands of the DNA have to unwind so they lose their spiral structure, and then the hydrogen bonds holding the bases, the two strands together between the complementary bases break down. Now each of the two original DNA strands act as template strands, meaning that activated nucleotides with extra phosphate groups are attracted to the exposed bases, and these bases have to be complementary so that they can form hydrogen bonds, where adenine forms bonds with two hydrogen bonds with thymine, and guanine forms three hydrogen bonds with cytosine, and the phosphodiester bonds are catalyzed by the DNA polymerase enzyme. So you're supposed to write the length of the entire process. 5 countries that have a high number of cases of malaria also have problems with diseases caused by bacteria. This means that many people in these countries are prescribed antibiotics such as penicillin for the treatment of bacterial infection. Now keep in mind that the pathogen causing malaria, which is a plasmodium, is not a bacteria, it is a protoctus, so it cannot be killed by these antibiotics. A. Outline how penicillin acts on bacterial cell. Penicillin inhibits the enzyme that is supposed to form crosslinks between the peptidoglycan molecules of the cell walls of these bacterial cells. And because peptidoglycan plays a great structural role in the bacterial cell walls, that means if the crosslinks between the peptidoglycan molecules are not formed, the cell walls get weak. And as water moves into these cells, it would swell and it would eventually burst, since the cell wall is not able to provide the support that it was supposed to do. The female Anopheles mosquito is the vector of the Plasmodium pathogen that causes malaria. The insect takes in Plasmodium when feeding on blood from an infected person, and at a later stage, the insect can transmit the pathogen when taking a blood meal from an uninfected person. These statements here provide a concise description of the transmission cycle, where the pathogen is transferred from an infected person to an uninfected person by the vector Anopheles. B. Name one of the 4 species of Plasmodium that can cause malaria. You can write anyone over here. I'll be going with whale. C. The male Anopheles mosquito does not feed on blood and so does not act as a vector. Suggest why there is a difference in the feeding behavior between male and female Anopheles. So male Anopheles do not produce eggs, and so they do not need a particular protein which female Anopheles mosquitoes do for the production of egg, and they get this protein from by taking blood meals. The research has shown that Plasmodium is not always transmitted to uninfected people. 2 main reasons for this have been suggested, and the first one says the immune system of mosquito kills Plasmodium while it is still in the gut, and the second one suggests that bacteria living in the gut of mosquito compete with Plasmodium, so it does not survive to continue its life cycle. A study was carried out to see if taking antibiotics affect the risk of transmission of malaria. Some observations and results of the study are summarized in figure 5.1. So the first flowchart says individuals with malaria are taking antibiotics, and Anopheles mosquito takes blood meal, and this leads to a higher survival rate of Plasmodium in the gut of Anopheles. Whereas in the second case, individuals with malaria that are not taking antibiotics have Anopheles mosquito taking blood meal from them. There will be a lower survival rate of Plasmodium in gut of Anopheles in this case. Suggest explanations for the results shown in figure 5.1 and comment on the importance of these results for doctors working in countries that have malaria and a high number of bacterial infections. Alright, so in this case, it was suggested in the question that bacteria living in the gut of mosquitoes compete with Plasmodium, so it does not survive to continue its cycle. That means bacteria in the gut of mosquito are essential for Plasmodium to be eliminated or killed, which is why you see that there's an increase in survival rate of Plasmodium in people in cases of malaria where antibiotics are taken, because these antibiotics would kill these essential bacteria and have no effect on Plasmodium. And if these bacteria are killed, that means there is no competition for Plasmodium in the gut of Anopheles. That means the Plasmodium would remain alive in the gut of Anopheles, and it would then be transferred into the next uninfected person that the Anopheles mosquito takes a blood meal from. Which is also why you see that individuals with malaria that do not take antibiotics, that means these antibiotics would not harm the bacteria of the Anopheles mosquito when an Anopheles mosquito takes blood meal and has the Plasmodium transferred, and so bacteria in that Anopheles would be surviving. They would compete with Plasmodium, and there would be chances of survival of this bacteria and them surviving to kill the Plasmodium. That leads to lower survival rate of Plasmodium. You'll have to explain that, and this means that for doctors, to reduce the risk of Plasmodium surviving the competition, what I read for the suggestion is that it is vital that they use antibiotics of a narrower range or specific to the diseases that they want to cure, and so they do not act against the bacteria living in the gut of mosquitoes. So they need to use antibiotics of narrow range spectrum. After many years of intense research and development, WHO reported in 2016 that a pilot vaccination program would be trialed between 2017 and 2020. The program uses a vaccine acting against the most widespread species of Plasmodium. Explain the difficulties faced by researchers in developing a malaria vaccine. For developing a vaccine for malaria, the greatest difficulties would be due to antigen concealment and antigen variation. So antigen concealment would occur when the pathogen in vaccine would live inside the host cells, and so an immune response would not be stimulated, therefore making the vaccine ineffective. Since the whole point of vaccine is to stimulate an immune response, to provide active immunity by producing memory cells and all of that. And coming to the second point, antigen variation. Because Plasmodium has many genes that code for many antigens, it's difficult to produce a vaccine that is specific to all of the antigens of the pathogen. Also, Plasmodium has many species, all of the 4 species that have their own antigens. So this also adds up to the difficulty in making a vaccine specific to all of the antigens. And a third point is about how the antigens of even a single species change when they go through the different life cycles in the human body. Six, telomerase is an enzyme that is important in maintaining the telomeres that are located at the ends of the chromosomes. Explain the importance of telomeres. So these telomeres are the repeated DNA bases that do not contain any significant information. They are only present so that when the enzyme that is supposed to be replicating the DNA, when they are not able to run the entire length and they leave these bases at the end, no significant information is lost. since these telomeres would be present at the end and they would not be replicated. So they ensure that genes are not lost, and this means that they are present throughout the cell's life. They can then be used again and again for replication. B suggests why telomerase is present in much higher quantities in stem cells and cancer cells than in any other cells. So cancer cells and stem cells divide more than all of the other cells, because in other cells the length of a telomere decreases every time they divide by mitosis, up until a point where the telomeres are lost and the significant parts of the DNA are also lost, and so they eventually die, which is not true for cancer cells and stem cells. They don't die. C studies suggest that telomerase is a flexible molecule when binding to its substrate. The active site changes its shape slightly to allow formation of enzyme-substrate complex. Name the theory that describes this mode of action. So an enzyme that has its active site that changes its shape to mold around the shape of the substrate acts according to the induced fit mechanism. D. An investigation was carried out into the effect of substrate concentration on the activity of telomerase. The results are shown in figure 6.1. So you can see that as you increase the substrate concentration, the rate of reaction increases. However, not only does it slow down after a certain substrate concentration, the increase in rate of reaction also decreases. With reference to figure 6.1, explain the difference in the rates of reaction obtained for telomerase with at a substrate concentration of 0.2 micromole per dm cube compared with the substrate concentration of 1.6 micromole per dm cube. So essentially, at a lower substrate concentration, the substrate concentration is the limiting factor, since there are a greater number of active sites present than all of the substrate. And so what happens is not all of them are occupied, only a few are occupied that form enzyme substrate complex with the substrates. As the substrate concentration increases, more and more enzyme substrate complex form. That means and at this point, the limiting factor would be active sites of the enzymes and not the substrate concentration. So that is all you need to write for this answer. And this is it. We are done with this paper. Thank you for watching.